the previous class, we discussed the cell cycle. Today we'll discuss it again in a detailed manner with some explanations so that you will understand better. I'm going to repeat the cell cycle, that is on page number 11. Take out page number 11.
Yes, ma'am.
Yes. So cell cycle 2.6, topic 2.6, cell cycle. That is divide, grow and redivide. Now you all know cells are present in every organism's body. So it must divide, grow and redivide. Why? Because we already know the bigger the organism, more are the cells. So our cells which are present, our life start as a single cell. But they have to continuously divide so that they can attain a mass so that they can attain a mass of cells and form tissues, right? Then these tissues form the organ systems, the organs and a proper organism at final, right? So all these cells must have to divide and then also grow and then redivide. Now you all know that the cell cycle has two phases. We have studied cell cycle in two phases, that is the interphase and the M phase. Now, before the cell cycle, before the starting of the cell cycle, the most important thing is to be done is that several important tasks are to be completed. What are that tasks? It is given in your book. That means the cells must grow first. They must grow. The second is they must synthesize proteins and RNA, right? And third one is the DNA have to be duplicated before splitting into daughter cells. Why? Why is the DNA have to be duplicated? Because when during mitosis, when they are formed into two daughter cells, both the daughter cells must get equal amount of DNA. So DNA has to be duplicated. So all these processes, that is these several important tasks, are to be performed before the cell division or before the cell cycle. Right? So you can underline this much. The cells must grow, synthesize materials like proteins, RNA, etc. and duplicates its DNA before physically splitting into two daughter cells. Underline this much. And this whole process, that means the growth, the synthesis of proteins and RNA and the duplication of DNA, all this is included in cell cycle. Right? All this is included in cell cycle. Now come to page number 12. Now it's written here, cell cycle consists of two phases. What are the two phases? First is interphase, second is the M phase, or we can say the mitotic phase, right? Now what is interphase? You have to understand the difference between two. So the interphase is basically the non-dividing phase. It's written in your book, you can have a look, underline or tick that it's a non-dividing phase. Whereas the M phase or the mitotic phase is the dividing phase. Now you will think interphase is a non-dividing phase, then why it is there? Division is not occurring, then why it is inside the cell division? Why it comes under cell cycle? It comes because interphase is a non-dividing phase. It is also known as a resting stage, right? It is also known as a resting stage. But during that phase, the cells are the cells are not properly resting. They are doing some work. What is that work done by the cells? That means they are synthesizing proteins and the RNA required for the next cell division, right? That means they are preparing themselves. For example, we are going for an annual function. We are the participants. Before the dance, we just prepare ourselves. We practice the dance. Like that, in this phase, interphase, the cells are not actually in resting phase. It is known as the resting phase before, but the cells are not actually in rest. They are synthesizing proteins and RNA for the next cell division. Which is the next cell division? The mitotic phase, where the cells actually divide. Right? So come to interphase. Here what happens? Two daughter cells are produced. Two daughter cells are produced in the phase. That means there's one parent cell, parent or mother cell. From that, two daughter cells produces, each having DNA. same number of DNA molecules. And during one more thing, during interphase, the chromosome number remains same. They don't get more or less. They remain same during the interphase process. Right? So this much is about interphase, which is the non-dividing phase, but actually the cells are not resting. What they are doing? They are preparing themselves, they are synthesizing proteins and RNA for the next cell division, for the next phase. Before entering into the mitotic phase, they are preparing themselves. Yes. So in interphase, the cells are actually not resting, but they are preparing themselves for the next cell division. Now, during interphase, as I have already told, the mother cell divides into two daughter cells, each having same number of chromosomes. The chromosome number does not vary. They both have duplicated DNA and they form a full-size nucleus. And we have already told that they prepare themselves for the next cell division during the interphase. Right? Now we will study detailed about interphase. Interphase is divided into three main phases. That is the first growth phase, first one. Second one is the synthesis phase. And the third one is second growth phase. That's on page number 12. You can see first growth phase, synthesis phase and second growth phase. So what is happening during the first growth phase? It's written in your book. The RNA and proteins are getting synthesized. As we all know, they are preparing themselves, the cells are preparing themselves for the next cell division. So the first point will be itself that they are preparing or synthesizing RNA and proteins. Right? They are synthesized in the first growth phase. Now second point is either the cell in G1 phase enter resting phase or start preparing for the next cell division. Right? So there are two possibilities. Either is there. There are two possibilities. Either the cell will go into resting stage, but there also it is not properly resting, right? It is doing some work there. It is synthesizing proteins, right? Or either they will go into proper rest. That means it depends on the cell. It is a possibility that either the cells will go into proper rest or they will go into a stage where they are preparing themselves for the next cell division. By what? By synthesizing the proteins and the RNA, right? So two possibilities are occurring here. You have understood the two possibilities. Either the cells go into rest or they are entering into the stage where they are preparing themselves for the next cell division. Then comes the next stage, which is the synthesis stage. So what happens here? In the synthesis stage, DNA gets synthesized. That means more DNA is produced, more DNA molecules are synthesized. Why? Because DNA will be required in the next phase. So more DNA is getting synthesized. Also, the chromosome which is present are duplicated. Right? They are duplicated. That means they are getting more in number for the next cell division. Right? So the chromosomes get duplicated and DNA is synthesized in a more way, in a more manner. This happens in the synthesis phase. So these are known as the first growth phase, that means G1 phase. Synthesis phase is named as S phase. And the second growth phase is named as just G2 phase, right? You can see second growth phase, G2 phase. Now what is happening in the G2 phase? RNA and proteins necessary for the cell division are being synthesized. So basically in the first G1 phase, in the synthesis phase and the G2 phase, in all these three phases, the RNA and proteins are getting synthesized for the next cell division. Because for the cell division, these proteins are required, right? Why they are required? We have already studied that earlier, that these proteins bound together, they coil around histones, right? they coil and they form a chromosome later, right? During cell division, when cell division occurs, they form the chromosome with the help of these proteins. So the synthesis of proteins and RNA is very necessary, it's a must. Now then again, cell enter the dividing phase. Now when these cells, after synthesizing RNA and the proteins, they enter the dividing phase. Which is the dividing phase? Can anyone answer? The name of the dividing phase. Can I get an answer?
M.
Yes, M phase. Very good, Priyadarshana. It's M phase, that is the mitotic phase. M phase is nothing but the mitotic phase. So these cells, after synthesizing RNA and proteins, they are entering into the division stage. That means the dividing phase, that is M phase. Then after entering into the dividing phase, they are forming new cells. Through division, new cells are formed. Then again these cells, these newly formed cells, they are again entering into which stage? The interphase. That means this process continues like a cycle. You know, you must have seen a cycle, a web cycle you must have seen. That's a cycle, that process continues, right? So similarly here also, after synthesizing RNA and protein, the cells are entering into the dividing space, right? There they are forming new cells through division, right? Then they are again entering into interphase. This goes on like a cycle. Then they are again entering into the interphase again, and the cycle continues. After again entering into the interphase, what happens? Then again from the start, G1 synthesis and G2 phase, and then again they are entering into division phase, M phase. Then again interphase. Then G1 synthesis, G2, then again division phase, M phase. Like that, like this cycle continues. So you can make a flow chart. You can make a flow chart like, on the top you can write cells synthesize RNA and proteins, right? When I'm saying, write this down in your notebook, right? You can make this flow chart. On the top you can write cells start synthesizing RNA and proteins in the interphase and synthesis stage. When the interphase stage के बाद they are entering synthesis stage, वहाँ पे भी RNA and protein synthesize हो रहा है. So you can write for your better understanding that cells start synthesizing RNA and proteins. Then cells enter the division phase. In brackets you can write M phase or the mitotic phase. Then put an arrow. You can write new cells are formed. New cells are formed. Right? After entering into the dividing phase, new cells are formed. Then put an arrow. Then you have to write the newly formed cells enter into the interphase again. The cells which are newly formed, they are entering into the interphase again. And then after that, one more arrow, and you have to write the cycle continues. That means when these cells are again entering the interphase, then they are starting from the start itself. They are starting from the beginning. That means the three stages in interphase: G1 synthesis and G2. G1 synthesis and G2. Then after from G2, they are moving to mitosis phase, mitotic phase, which is the division phase. Then the cycle again continues. Like this, cell division occurs in an organism. Am I clear to you all?
Yes, ma'am.
Yes.
Yes, ma'am.
If you have any doubt, you can ask me right now regarding the interphase and the entering of cells into the mitotic phase. This much, is it clear? If you have any doubt, you can ask.
No doubt, ma'am.
Okay. So you prepare a flow chart. As I've told you, prepare a flow chart. You can have a look in the book also. After this class, you can prepare a flow chart by yourself also. Very easy to understand. Now come to, yes, so the second growth phase we have done. Now come to page number 13. So this formation of new DNA, can the cell cycle go endlessly? We have discussed this earlier, and it is not that important. You have to just go through this paragraph just once, how the formation of DNA is taking place. Now come to cell division, which is on page number 13. Page number 13, cell division. So, as we all know, there are two types of cell division. Which are they? The mitosis and meiosis. two types of cell division, mitosis and meiosis. Now what is the difference between the both? Now mitosis division leads to the formation of diploid cells, which helps in growth and formation, growth and development. That means diploid means one parent mother cell is present and from that mother cell two daughter cells are produced. This is mitosis, right? And whereas in meiosis, the formation of haploid cells is occurring. Haploid means first from the parent cell, the two daughter cells are produced. Then these two daughter cells, that means this one and this one, they further divide to form two more. That means this one is forming two more and this one is also forming two more. That means at last there are four cells in total. Four daughter cells are formed. That means haploid. Haploid means four, more than two, right? And diploid means two. So similarly, as mitosis we know two daughter cells are formed, so diploid cells are formed which helps in growth and development, and in meiosis haploid cells are formed, or we can say gametes like sperms and egg, sperms in male and egg in female, right? Gametes are formed as a result of meiosis. So basically in humans, which of the cell division is occurring mostly? Meiosis, because it is forming gametes, haploid cells are formed, right? Now I just come to page number 12 again. You can see the diagram on the top, cell cycle. Okay, G1 phase, S phase, G2 is given, right? Cycle is shown. What is happening in G1 is shown. RNA and protein synthesized, mitochondria is getting divided, chloroplasts are dividing. Then in synthesis phase, more DNA is synthesized and the chromosomes get duplicated. And in third G2 phase, more RNA and protein is further synthesized for the cell to enter into the next division stage. So then after this G2 phase, they are entering into the division stage. Now you can see mitosis, cytokinesis, karyokinesis, right? This all comes under the division phase. That means mitosis is further divided into two phases. That is, first is cytokinesis and second is karyokinesis. And that we will learn in the next topic. So this comes under the this purple portion with the labeling mitosis, cytokinesis, karyokinesis, and the prophase, metaphase, anaphase, telophase. Everything comes under division stage. That means the M phase. And before that, the second growth phase, synthesis phase, first growth phase, this comes under interphase. So you can just label it like for your better understanding, you can label these three as interphase, like we have written here, resting phase, and then on the upper side you can mark that and write the M phase or the dividing phase for your better understanding, right? Now cell division, we have talked about cell division, two types, mitosis, meiosis. Mitosis gives out diploid cells, meiosis gives out haploid cells, right? Now come to mitosis. So, as we have already said, in mitosis, same number of chromosome number is maintained. Means during the entire cell division, same chromosome number is maintained in that. No less, nothing less or more like that, but the same number is maintained during the division process. Now, phases of mitosis. I told you mitosis have two phases, two steps, karyokinesis and cytokinesis, right? Karyokinesis and cytokinesis. Karyokinesis means division of nucleus. Now, how you can learn this? You all know that nucleus, scientific name, we can say nucleus as karyon, right? Karyon refers to nucleus. We have earlier studied prokaryotic and eukaryotic. In that, karyon means nucleus, right? So this nucleus, division of nucleus is known as karyokinesis. Now what is cytokinesis? From the name itself, it is very clear. Division of cytoplasm. Cyto means cytoplasm. So division of cytoplasm is known as cytokinesis. Now you must be thinking, why this only this nucleus and cytoplasm is dividing? Why not the other organelles? Why? Because both of these, the nucleus and the cytoplasm, both of them are the living substances in the cell. Be it a plant cell or an animal cell, the nucleus and the cytoplasm both are the living substances. And you all know what are they together said? Can anyone answer? Nucleus and cytoplasm are together referred as protoplasm, right?
Protoplasm.
Yes, protoplasm. You have studied in this chapter or maybe in ninth, I don't remember. Yes, it may be in ninth standard you may have studied that nucleus and cytoplasm, both of them together are known as protoplasm. So basically protoplasm is the living substance. Protoplasm consists of nucleus and cytoplasm. Both of these are living substances. So they are getting divided, right? Because living substances का division होगा तभी तो आगे जाके cells बढ़ेंगे. Non living substances का division करके क्या मिलेगा? Nothing. So living substances are getting divided. Now we have known that karyokinesis is the division of nucleus, cytokinesis is the division of cytoplasm. Just a minute. Yes. So karyokinesis of mitosis occurs in four main phases. Now this karyokinesis, that means the division of nucleus is occurring in four main phases. What are they? Prophase, metaphase, anaphase and telophase. Prophase, metaphase, anaphase and telophase, right? Yes. Now, before that, this mitotic phase, we have told that karyokinesis and cytokinesis is occurring. Both of them together form two identical daughter cells. Like in the outcome or the result is production of the two identical daughter cells, and the DNA replicates in them. The DNA replicates means it forms a copy. It is forming a copy. So at the last, after this mitotic phase, that is karyokinesis and cytokinesis, the outcome or the result of both of these are that they produce two identical daughter cells. Now we know karyokinesis is divided into four: metaphase, prophase, metaphase, anaphase and telophase. So in some cases, prometaphase is also present, which is the mixture of prophase and metaphase. But you have to study four here. So first is prophase. Now what happens in prophase? First point, I am reading this on page number 13, prophase, first point. The chromosomes have become short and thick and are clearly visible inside the nucleus, right? So in prophase, pro means the first phase. What happens here? The chromosomes become short and thick. Nucleus is very clearly visible and are thick and are clearly visible inside the nucleus. That means chromosomes are present where? You have seen in the diagram, inside the nucleus, chromosomes are present. Actually, you know, inside the nucleus, chromatin threads are present. These condense themselves during cell division. They convert into chromosomes. So, from that we can make out one point, that chromosomes are very short and thick during this first phase. They are very short and thick and are clearly visible inside the nucleus. They look like short threads, right? Each chromosome has already duplicated to form two chromatids. Now we all know in cell division, when chromosomes are formed, one chromosome is individually known as chromatid. We call one chromosome individual, one chromosome we call it as a chromatid, right? So each chromosome has already duplicated. Why? Because this is the dividing stage. In the first phase itself, the chromosome has been duplicated. So this duplicated chromosome, the individual, it will be known as chromatid. That is what is written here. Each chromosome has already duplicated to form two chromatids. That means one single chromosome. Chromosome looks like an X shape like this. So both of them are divided. That means they are single now, right? So this one is one chromatid, this one is the second chromatid, right? So they're individually known as the chromatid. Now the two sister chromatids remain attached to each other at a small region called centromere. So you all know, you can see you can imagine the diagram of a chromosome, or you can take out page number 10. The diagram is given on the top, chromosome diagram is given, right? So you can see in this, a middle portion is there. You can see a portion where two chromatids are crossing each other. This middle portion, that means the middle portion, this middle portion is known as centromere, right? So that is what is written. The two sister chromatids, that means these individual chromatids, they are attached to each other. They are attached in the point known as centromere, right? Fourth one, the centrosome in animal cells splits into two along with simultaneous duplication of centrioles contained in it. Now we all know animals have centrosomes in it. Plant cells do not have centrosomes. So the animal cells, the centrosomes present inside the animal cell, they start splitting into two, right? The centrioles start splitting into two. The daughter centrioles move apart and occupy opposite poles. Now we have already studied, maybe in this chapter only, yes, this chapter, the next page, it is written that the daughter centrioles, the centrioles which have divided, which have been splitted, from these centrioles, one of the centriole occupy the top pole. That means inside a chromosome, they occupy, the one occupies the one pole and the second goes to the downwards pole. So they both occupy one pole each, right? So they form an aster, a star-like figure after entering into the poles. After occupying space in the poles, they form a star-like structure which is known as aster, right? This is only in the case of an animal cell, because centrosome is present only in animal cell, right? A number of fibers appear between the two daughter centrioles, which are called the spindle fibers. Now these centrioles have number of fibers which are known as spindle fibers. You all know these are spindle shaped, that's why they are known as spindle fibers. The nuclear membrane and the nucleolus disappear. Now during this prophase, the nuclear membrane and the nucleolus gets disappeared. The duplicated chromosomes start moving towards the equator of the cell. What is an equator? Equator is the middle portion. Like if we see the map, like a total globe we can see, so equator is in the middle, right? So after this process, the duplicated chromosomes, the chromosomes which are duplicated, which are now in the form of chromatids, they start moving towards the equator. That means they collect at the middle portion. So on the next page, 14 number, a diagram is given. We will have a look at the diagram later. First we will discuss the theory, right? So all these chromatids form, the two chromatids, the sister chromatids, the duplicated chromosomes start moving to the equator, to the middle portion of the nucleus, right? This happens in the prophase. What all is happening? Chromosomes are thick and visible inside the nucleus, right? They get divided and they are known as chromatids. Now these chromatids are attached at one point known as centromere, right? Then in animal cell, centrosome splits the centrioles into two or more, and they form, they occupies two poles each, sorry, one pole each. That one goes to the one pole and the second goes to the downward pole. They form a star-like structure there. That star-like structure is known as aster, right? Then nuclear membrane and the nucleolus gets disappeared, and at last what happens? The chromatids or the duplicated chromosomes formed, they move towards the equator or the center of the nucleus. That's it. That's all about the first phase, which is the prophase. All these things happens in the first phase. Now comes to metaphase. Meta means after. That means after the prophase. First phase is the prophase, then after phase. The duplicated chromosomes arrange on the equatorial plane. Each chromosome gets attached to a spindle fiber by its centromere. Now what happened at the last in prophase? The duplicated chromosome is occupying the center, are moving towards the equator. Now these chromosomes in metaphase, they arrange on a equatorial plane. That means on the equator they arrange themselves. And then each chromosome is attached by a spindle fiber by its centromere. You all know centromere is the point where the chromatids are attached. So each chromosomes get attached to the spindle fiber by its centromere. That means through the centromere portion, the chromosomes get attached to the spindle fibers which are present inside the nucleus. Right? That means they are just threaded around. They just coil around. Now come to page number 15. We will discuss this diagram later. This is just for your better understanding. So come to page number 15. Third one is anaphase. After metaphase comes the anaphase. Now what is anaphase? Ana means up, back. Pro means first, meta means after, and ana means up or back. The centromere attaching the two chromatids divides and the two sister chromatids of each chromosome separate and are drawn apart towards opposite poles. The drawing action is performed by the contraction of spindle fibers. Now in anaphase, what happens? Again, the chromatids which are there, which are arranged, the chromosomes are arranged in the equatorial plane, they are again taken back or they are drawn apart to different poles, right? They reach the same position from where they came from. They were in the different position, different poles in the prophase, right? Then in the metaphase, they just, just in the last point in prophase, they comes to the equator. They form a line in the equatorial plane. And then in anaphase, again what is happening? They are again drawing themselves apart to the different poles. The drawing action is performed by the contraction of spindle fibers. Now this drawing action, that means this, when they are moving apart to different poles, this action is performed through the spindle fibers. These thread-like fibers helps these chromosomes or the chromatids to move apart. Then fourth one is telophase. Each chromatid or the daughter chromosome lengthens, becomes thinner and turns into a network of chromatin threads. Nuclear membrane reappears, nucleolus reappears in each daughter nucleus. So what is happening here? We have already said that interphase के बाद the cell goes into the dividing stage and then the cycle continues, right? So what is happening here? Telophase में जब आ जाता है this thing, each chromatid or the daughter chromosome, ये फिर से lengthen हो जाते हैं, they become thin again. Okay, they become thinner and turns to a network of chromatin fibers. जहाँ से वो शुरू हुए थे, like chromatin threads, they condense and form the chromosome. And after entering into the division phase, they are again entering into the interphase again. That means the resting phase, right? So they again, they gain their normal shape and size in telophase. That means the chromatids or the chromosomes, they become thinner and seems like turns into a network of chromatin threads. Now again the nuclear membrane reappear हो जाता है. Nucleus also reappears when it is when these both of these are just disappearing in the prophase. In the prophase, nuclear membrane, nucleolus disappear हो जाते हैं. But in telophase, they are just reappearing again, and they are occupying or they are gaining their first, first structure. That means from where they started, the chromatin threads. So as we already told, the cycle is continuing. After the division phase, they are entering into interphase again. So they are gaining back their normal structure, which is the chromatin threads. All the contents of the cytoplasm, including the mitochondria and the chloroplast, are randomly distributed in the daughter cells during karyokinesis. Now, during this division of nucleus, all the cytoplasm, including both the mitochondria and the chloroplast, everything is distributed in the daughter cells when during the division of nucleus. Now come to page number 14. Come to page number 14. It's just for your revision. You can see interphase, resting phase. Okay, no changes visible. Why? Because they are in rest stage. Some cells have gone to rest, some are preparing themselves for the next stage. So no changes visible here. Then prophase. You already know, we have studied prophase. Centrioles moves apart, they occupies different poles and they form a star-like structure, aster, right? And nuclear membrane and nucleolus disappear, right? This happens in the prophase. And at last what happens? All the chromosomes formed, comes into the equator, right? Then enter into the metaphase. Now here each chromosome gets attached to spindle fiber by its centromere. Chromosomes line up in one plane. They are forming a line in the equator. Then they are entering anaphase. What is happening in anaphase? Centromere attaching the two chromatids divides. Now the centromere attaching the chromatids, it gets divided. Then two sister chromatids, then again they are drawn apart to different poles. That means what happened in the prophase, they were in different poles first. Again in anaphase, they are aparting to different poles. Now these fibers, these chromatin fibers get thinner and thinner. So in telophase at last, these get very thin. The chromatin fibers, these get very thin. Nuclear membrane is again reformed. It is again reappearing. Also the nucleolus is again reappearing. The cleavage furrow starts deepening in the animal cell. Nucleoli reappear. All these we have discussed in the paragraph itself. So this is just for your better understanding. In points it is given. So you can learn from here. Now you can go through the diagram. This diagram is all what we have said right now. Right? So first diagram you can see animal cell, centrosome containing centrioles, cytoplasm is there, chromatin threads is there. Now in the second stage, the chromatin threads, they are condensing into chromosomes. You can see chromosomes here, right? Between them centromere is present, nuclear membrane is starting disappearing. So if you learn, try to learn this with the help of these diagrams for better understanding. If you try to grab things or if you try to learn these long paragraphs, it will be very difficult for you to understand things. But if you go through the diagram, it is very simple and clear in the diagram. See, in the third diagram in animal cell, they have formed an aster in different poles, right? And then in metaphase, chromosomes lie along the equatorial plane, right? They have formed a plane, a line is formed. Then again the sister chromatids are differentiated. They are moving toward different poles, right? In late anaphase, a furrow-like structure is formed. That means a bent is formed. And in plant cell also you can see plant cell ka process is little simpler than the animal cell, right? Chromatin threads are there formed into chromosomes later. Then they are moving to different planes. Again same thing is happening. They are forming a line in equatorial plane. Then they are again dividing. They are aparting themselves to different poles. And then finally they are at the different poles again. They are again regaining their normal structure, the chromatin threads. So that's the whole thing about karyokinesis, right? Karyokinesis, cyrokinesis, everything. Cyrokinesis is the cleavage furrow deepens totally in animal cell and separates the two daughter cells. That means in animal cells, the cytoplasm gets divided and a furrow-like structure forms between the cell, the animal cell, and the two daughter cells. They get divided, like they divide totally. You can see the example. You can see the diagram in the page number 15. Okay, animal cell is there. A furrow is formed at the last anaphase, and these are dividing into two daughter cells, each with chromatin threads, nucleus, and asters in them. Right? So this is all about karyokinesis and cytokinesis, together known as mitosis. Right? So we will discuss this differences and all the other portions in the next class. Do you all have any doubt till now? Anyone?
No doubt, ma'am.
No doubt. So you have understood the process. It is simple. If you go through the diagram, you will get it easily. Okay, try to understand the diagram more promptly. So you will be able to write this in exam in a more proper way, prominent way, right? So that's all for today. Thank you everyone for joining. Okay, we will meet in the next class. We will discuss the next topic in the next class.